brain regions, and some of their associated visual behavior. Without the whole retina, we cannot see. Our visual acuity is much better in a central visual field where there is a much higher density of retinal neurons. Central vision loss can be caused by, for example, macular degeneration. It typically affects older people. It makes it difficult to do tasks such as reading and face recognition. Tunnel vision is caused by a loss in peripheral visual field, for example, by glaucoma. It makes navigation difficult, and depending on situations, this often affects life quality much more than central vision loss. We naturally have a blind spot in each of our retina, and this is the spot called the optic disc, where the axons from the retinal ganglion cells exit the retina to project to the central brain. This blind spot is small enough, and the blind spot in one eye can be compensated by normal vision in the other eye so that its effect is typically not noticeable. Damage to the primary visual cortex, V1, makes a person clinically blind. However, there is some residual visual abilities called blind sight. Although patients without V1 claim that they cannot see, somehow they can localize or navigate around an obstacle or even discriminate it somewhat. If you damage V1 in a monkey, it takes 2 months before the monkey can succumb to a bright spot flashed on a blank screen. It is likely that the brain eventually rewires the neural circuit to some extent after V1 damage. Let's recall that the ventral visual pathway is more associated with object recognition for what an object is. In comparison, the dorsal visual pathway is more associated with where something is for reaching or grasping. And here we show a demonstration for the what and where tasks. The what task is to hold a plate in hand so that its orientation matches the orientation of a post slot, but there's no need to insert the plate into the slot. So this task requires recognizing the orientation of this post slot and reporting this orientation by holding the plate correctly. And the other task, the where task, is to actually insert the plate into the post slot. So this requires an action without verbally or consciously reporting the orientation of the slot. And here we show the orientation of the plate held by a patient to do the matching and posting tasks. This patient has damage to her ventral visual pathway. Her orientations are quite random when doing the what task, but quite accurate when doing the where task. For normal people without brain lesions, the plate orientations are quite accurate in both tasks. And this suggests that the brain mechanisms for doing the where and what tasks can be dissociated from each other. Hemi-neglect is a deficit of attentional awareness to half of the visual field after damage to one hemisphere of the brain, most commonly associated with damage to the right hemisphere. So for example, a damage to the right parietal lobe, and then as a consequence, the left half of the visual field is neglected. For example, if the patients are asked to find all the letters A in here, they might just circle these instances and not the instances in the left half of this image. If patients are asked to draw a clock, this might be an outcome. This is apparently a damage associated with the spatial frame of reference in attention. Somehow the left half of the circle is drawn after all. After the onset of a visual input, V1 neurons start to respond about 40 milliseconds later. For V4, it takes about 70 milliseconds before they start to respond. and this is 100 milliseconds for IT neurons. By 125 milliseconds, responses from 300 IT neurons can be used as a population to classify visual input objects into categories by a linear classifier. However, when object discrimination is harder because, say, some parts of the objects are occluded in the visual inputs or are deformed, then we need to wait for later parts of IT neural responses to use them in a linear classifier to classify objects sufficiently well. And this is both in monkeys and humans. And this temporal delay suggests the involvement of both feed-forward and feedback processes in the brain's neural circuit to achieve more informative neural responses.